Thank you for joining us today for this Physical Sciences Oncology Center mini symposium on the fundamentals of cancer metastasis. We're very pleased to be able to host this type of interaction. The Northwestern Physical Science Oncology Center is really a very unique experiment by the National Cancer Institute to bring physical science perspectives from the fields of mathematics, applied math, modeling, physics, and engineering to apply to problems and to really delve down and obtain a deeper understanding of origins of cancer, its evolutionary context, if that makes any sense, and we debate that all the time, and ranging all the way through the physical and mechanical studies of the polymers in the cell that ultimately, when changed, cause initiation of a cancer phenotype. The importance of these types of studies is, in my mind, the key to discoveries that will come in the next 10 and 20 years in the central fields of cancer biology, where we might actually see applications of the discoveries to development of new therapeutics or to the development of early stage diagnosis. The ideas generated here, therefore, are the kind of the seed corn, and I think it's really an exceptional symposium that Bennett Phillips has organized for us that brings together some of the best cancer biologists at Northwestern to help educate the physical sciences community. And I say best scientists at Northwestern, even though we imported a ringer from Indiana, from that little football school down south. I can't remember the name of it, but actually it's Sharon Stack, and Sharon is actually a former Northwestern faculty member who is a very active member of the Cancer Center and a very important contributor in the field of understanding invasion and angiogenesis. And we have a talk by her today. But I thought at this point maybe I'll ask Bennett to say a few words about the program that she helped put together.
Okay, so I will mention that this is actually our third summer workshop on the fundamentals of cancer. And the first one dealt just with basic cancer biology. Last year we had a workshop on cancer therapeutics, successes and failures. And this year I decided the workshop would focus on metastasis. I mean, and for really, there's two basic reasons. Well, there's probably more than two, but two that came to my mind are, number one, it's actually an extremely complex biological process, and I mean, if you have to put an adjective to it, it is biologically, it's fascinating because these cancer cells can do, you know, just they go through an amazing series of steps in order to form metastases. The other side of it is it's extremely deadly for the patient to develop metastatic cancer. We really, unfortunately, there's, you know, the treatments can retard the disease, but most people succumb to the metastases and not to the primary tumor. So we're going to have four speakers. The first two are really going to kind of focus on basic mechanisms of metastasis. The third speaker is going to talk about animal models because they're absolutely invaluable in studying this disease. It's very hard to study this in isolated cells. You really have to look at it in the context of the whole organism. And then the last speaker is a clinician who's going to talk to us about management of patients with metastatic disease. So I'm certainly, you know, glad to see, as usual, a nice sized audience, and I hope that you enjoy the talks, and there'll be a period for questions after every talk, so we hope that you're going to, you know, participate in the question and answer period. I'm sure all the speakers are going to welcome questions, and as you know, this workshop does not presuppose that you have any background in cancer. Some of you may have just minimum, you know, exposure to biology even, so don't be afraid to ask questions. So our first speaker is Carol LaBonne, who's actually housed up on this campus. So she's associate professor, right, in their full professor? Huh? Till September. Okay. So, and the Department of Molecular Biosciences, and her interest in metastases, I think, is because of its similarity to a process that occurs during embryogenesis, where cells are able to detach from the neural crest, and they undergo something called an epithelial mesenchymal transition, which also happens in cancer, and they acquire these migratory properties, and they can migrate away from the neural crest. I'm telling this right, and form all and form, because this is not my area, and form a wide variety of specialized cells. So she's going to start out and talk about the epithelial mesenchymal transition and some of the factors that are responsible for this. So I will turn it over to Carol.
You were doing a great job. So I kind of get the fun part of this, and then I get to do the introduction. Is that too loud? I think we need techno guy here. Oh, you know what? You left this on, maybe? No? Can you hear me? Okay, let's just stick with that. I'm fine with it. Okay. So I get the fun part of sort of giving you a historical perspective and introducing you to the sort of the concept and the biology and the history a bit of EMT. Before I start, I wanted to point out, in addition to what Bennett and Tom told you, that all four of the speakers, the current and our one former, are a part of the Cancer Center, which is really a fundamental part of the basic science division of the Cancer Center. We call it TIMA, or Tumor Invasion, Metastasis, and Angiogenesis. And it really is one of the most exciting areas of cancer biology these days, and the one that in many ways might be the most therapeutically promising. And so hopefully from both my introduction and the three talks that will follow, you'll really get a good sense of why it is that this is such an exciting area of biology and such an important one in terms of the disease process. So as I said, I'm going to give you an introduction. I am one of the two co-leaders of TIMA, along with Kathy Green downtown. I'll be followed by Sharon, who used to be the co-leader of TIMA with me before she abandoned us and went off to do great things elsewhere. And then the two other speakers are also part of this division of the Cancer Center. Okay, so I really want to take you through the basics of cancer biology a bit again. And this diagram is drawn from a really seminal review in Cell by Bob Weinberg, who's a professor at MIT. Came out about 10 years ago. There's actually a more recent version that came out about a year ago, although I like the original one a little better because it's simpler. And really, cancer is complicated enough without adding on even more bells and whistles beyond the basics. But what this basically shows you are the kinds of things that a regular, respectable cell in your body has to change in order to become a tumor cell, and then in order to become an invasive tumor cell. And that's an important distinction there, because as Bennett alluded to, most cancer deaths are not from primary tumors. Over 95% of cancer deaths are from metastases. And so that distinction between acquiring tumorigenic features and then becoming an invasive tumor cell is an important one. But there are a number of things that a cell has to be able to do. Okay, it has to gain self-sufficiency and growth. It has to become insensitive to anti-growth signals, to signals that would tell it to stop growing. It has to be able to avoid undergoing apoptosis, which is what your normal cells are supposed to do if those processes of growth and anti-growth are dysregulated. It has to overcome cellular senescence. Most times cells in your body can only replicate a certain number of times. Tumor cells have to overcome this so that they can gain what we call cell immortality. Okay? And so all of these things together allow a tumor to grow. Once it reaches a particular critical size, it has to do one more thing in order to keep growing, which is to attract the vasculature so it continues to have a source of food and oxygen. So angiogenesis, I mentioned, was an important part of that TIMA, and it's an important part of tumor growth in biology. And again, these things are what's going to give you what we call a carcinoma in situ. Oops, I'm going too ahead of myself. It's too many slides ahead, so I'll come to in a second. And then you need this one additional thing to acquire the ability to leave a primary tumor and go elsewhere to form these new secondary tumors or metastases in order to become an invasive cancer. And then again, that is where most cancer mortalities arise. This is important when thinking about therapeutics, and I know a number of you are interested in that, and I'm going to close on some therapeutic data as well today. But historically, the things that have been developed thus far as cancer therapeutics have been screened by their ability to shrink or block cancer growth. Okay, so they're going to be interfering with most of these things, except for the one that we're going to be focusing on today, including to some, as there's been some beginnings of promising angiogenesis inhibitors, although as I'll mention towards the end, so a major problem with drugs that are targeting these other events is that they may prevent cancer growth, but they don't get at something that we consider to be super important, which are cancer stem cells. And these are the cells that are going to have the ability to seed those new cancers of metastases. And so although you may block tumor growth, you're going to have recurrence. Okay, and one major problem with angiogenesis inhibitors has been that it tends to make tumors hypoxic, and recent literature has suggested that tumor hypoxia actually promotes EMT and metastasis. So again, while you may in fact block tumor growth, you may in fact be seeding the process by which those more dangerous metastases form. And so a theme that I want to get back to at the end is that a really promising place for being able to intervene therapeutically that might have high clinical relevance in terms of outcome is going to be in blocking the process of metastasis, perhaps by blocking it early in this event called EMT. Okay, so this is just summarize what I told you in terms of therapeutics. Really, many of them have been screened literally by their ability to shrink tumor size in animal models. Okay, and so what that's going to do is stop these transient amplifying cells that are going to cause tumor growth, but it doesn't get at these few cells that people are beginning to believe are the cells that are going to have the capacity to make those new tumors at remote sites. And so then you have the ability for those cells, after a period when the therapeutics are no longer present, to come back and make these new tumors grow. Okay, so then tumor invasion and metastasis is a process by which those cells that have undergone all those changes that I mentioned from von Warburg's nice slide, okay, where they now acquire one more trick in their arsenal, and that is the ability to lose contacts with their neighbors, to be able to remodel the matrix and in fact invade into the vasculature, to move elsewhere in your body, and I think Jennifer's probably going to tell you about breast cancer cells moving to bone, and then to form these secondary tumors or metastases, which is where, again, more than 95% of cancer death occurs, okay? And so that really is considered to be a four-step process, okay? Molecular changes that happen within that tumor cell that allow the acquisition of invasiveness, and I'm going to spend some time talking about that today, because a major step of that is believed to be this process that Bennett mentioned, which is epithelial to mesenchymal transition, okay? And then there's intravisation, or entering into the vasculature, okay? The ability to leave the vasculature at target sites where tumor growth is favorable, okay? And then a process that's actually really understudied, but really super important, the opposite of EMT, MET, mesenchymal to epithelial transition, because you're going to have to form again an epithelium in order to make that new tumor at a remote site. Okay, so I mentioned I want to take a bit of a historical perspective, and so what I'm going to kind of walk you through for the beginnings of my talk are several major discoveries that had to be made and then come together to bring us to our current understanding of EMT and metastasis. And the first was made by a woman named Betty Hay, who was a mentor of mine. She was actually the chair of anatomy and cell biology at Harvard when I was a graduate student, and was the person who first taught me about EMT. But she was the person who coined the term and first described it, and she was remarkable for that reason, but for a couple of others as well. She was the first full professor in a non-clinical department at Harvard Medical School, so she was really a pioneering woman in science, and one of the first female department chairs in an Ivy League school as well. So she was really a towering figure academically at the time for many reasons. In fact, she literally wrote the book on interactions between cells and matrices and wrote it repeatedly. And so she was a person who studied interactions between cells and interactions between cells and their environment and their matrix. And in the course of doing that, she made this really fundamental discovery, which was that cells that originate oftentimes in organs as columnar epithelia, both in organs in the adult and also in epithelial tissues that are developing in the early embryo, which is where Betty first made these observations. These cells have very characteristic features and connections between themselves that I'll go into a bit in a moment. But she was the first to observe that sometimes these cells could undergo a transition where they lost these characteristics of columnar epithelial cells and acquired a more mesenchymal phenotype. Okay, and so this is critical, it turns out, for many areas of development as the embryo progresses and develops a body plan, as organs form later in life, as tissues remodel, and then the process itself is reactivated in a number of diseases, cancer, which we'll be talking about today, but also in things like fibrosis. Okay, so there are lots and lots of EMTs in embryos. Okay, I'm gonna mention my lab studies. One, Bennett mentioned these neural crest cells that can migrate and invade through early embryos. The earliest event is one called gastrulation, where after you form the 3 germ layers that make up the building blocks of building an embryo body plan, they have to move with respect to one another to acquire the right positions, and they do that by having some of them undergo this transition where they lose contact with their neighbors and they can move more readily throughout the early embryo. And this is a chick embryo, which Betty used to study. And these cells here are cells that used to be part of this blue layer that underwent these transitions to move into the interior of this developing chick embryo. And this would look very much the same at a very early stage in human development as well. This is actually what it looks like in real cells. This is just a cartoon. And this is the cartoon of the process that my laboratory studies, which I'm not really going to talk to you about today, except to mention it briefly at the end. But at a certain point during embryonic development, another population of cells, which are a stem cell population, have to leave your developing central nervous system, which is also a columnar epithelial at that point, and again acquire the ability to invade and migrate throughout the body in order to give rise to many aspects of the vertebrate body plan, but a major portion of that is the peripheral nervous system. So they leave the central nervous system, invade and move very long distances in the embryo in order to then re-become an ectodermal cell that will contribute to the peripheral nervous system. And again, this is a picture of a real embryo with those cells seen here migrating into the periphery. So there are lots and lots of EMTs in the embryo. In fact, in the development of many organs, there have to be a succession of transitions from epithelial to mesenchymal and then back to epithelial. So it's a very, very important process in embryonic development, and that was recognized before the links to disease were realized. And again, there are characteristics of epithelial cells that have to be lost during this process and new characteristics gained by the cell population. Okay, so epithelial cells, columnar epithelial cells, have contacts with their neighbors called junctions, and these have to be disassembled or remodeled. Okay, mesenchymal cells lack those junctions. Instead, they have adhesion points with their underlying matrix. Okay, so focal adhesions that they have to be able to develop, which requires a different kind of membrane remodeling. There is different kinds of polarity in these types of cells. So columnar epithelial cells have something called apical basal polarity. One part of their membrane has physically distinct properties from other aspects of their membrane and is kept separate and regulated separately, and that's very important for their function. That kind of polarity has to be lost, and what these mesenchymal cells have is sort of a front to back polarity or a leading edge to trailing edge polarity that has to be established. And I have one characteristic here in red because it's not strictly true, although you'll find it in all your textbooks, where they tell you that epithelial cells are stationary, whereas mesenchymal cells are motile and invasive. This part is true, but lots of epithelial cells are not stationary. What is it that they move in one plane? Okay, they don't lose contacts with their neighbors and invade through other planes of tissue, but they can in fact be motile, and this is sometimes a problem with some types of experiments that are done where they look at cell motility and they think they're looking at invasive behavior when in fact they're looking at the kinds of behaviors that respectable epithelial cells sometimes undergo. Okay, there are also molecular markers markers. There are characteristics of each of these states, and that's very important for being able to follow them, whether you're following them in a developing embryo or following them in a developing tumor. Okay, so there are things, and I'll mention at one point E-cadherin. This is one of those molecules that helps cells stick together with their neighbors. Okay, that has to be lost from cells that are losing their epithelial characteristics in many cases, and then they'll gain expression of markers that are important for being a mesenchymal cell. Like Pimentin is a common one that people look at. So when Betty first coined the term EMT, she actually called it epithelial to mesenchymal transformation. And now we know that it's not so much a transformation as a transition. So we still keep EMT, but we call it an epithelial to mesenchymal transition, not transformation. And there really are in-between states in all of these things. Not all of those events I just mentioned happen simultaneously. And in some contexts where cells undergo these transitions, they undergo partial EMTs. They do some of these things and not others of these things. And again, there's a very important counteracting cellular state change, which is the MET, the mesenchymal to epithelial transition, which also can have partial aspects or complete aspects of the transition. And so many different kinds of EMT in an embryo, and not all of them look exactly the same, although they all share many common features. Okay, so the second historical thing I have to tell you then was, so first you had the observation, first in embryos and not yet even at this point in disease processes, that cells underwent these transitions. But then what we really needed was a molecular signature for them. We need to know how they undergo these kinds of transitions. And a lot of what we know about that came not from studies of humans or tumors, but from studies of very simple laboratory organisms like Drosophila. And so there were two people in particular, Yanni Nusslein-Volhard and Eric Wieschaus, who did massive genetic screens in the fruit fly and Drosophila to identify genes that were important for controlling various aspects of the body plan in a fruit fly. They ended up winning the Nobel Prize because not only were they able to do that, and at the time it was a heroic undertaking, and nobody really believed that you could use screens to categorize in some sort of a sensible way the major molecular players that build a body plan, but they not only did that for this simple organism, the fruit fly, but it was later shown both that those same molecules are involved in building the same analogous aspects of us as much more complicated organisms, and that those developmental control molecules were often misregulated later in life to mediate disease processes like cancer. And so this happens to show a Drosophila embryo undergoing that same process I mentioned, gastrulation, where cells change position with respect to one another to build a body plan. And this is a Drosophila embryo, but these dark cells you see that start out here at the bottom and then move inside are being marked by expression of one of the factors, one of the transcription factors that they isolated in these screens, and it's called TWIST. And TWIST, as it turns out, is a very important molecule, not just for gastrulation in Drosophila, but also for gastrulation in humans and for neural crest migration and many other developmental EMTs, and also for fundamental aspects of metastasis of almost every carcinoma that's been examined. Okay, so I can tell you another interesting historical context in addition to the fact that a lot of what we know about these genes that regulate cancer was learned in fruit flies. And that is, remember I mentioned that two people were really pivotal in this work and won the Nobel Prize, Yanni Nusslein-Volhard and Eric Wieschaus. They set up a laboratory together in Germany in order to do this screen and to make these discoveries. And part of that was due to the fact that they worked well together, and and this was really a heroic undertaking that took a lot of person hours in order to visually screen through mutant embryos looking for effects in phenotype. But the other reason why they did it was because Yanni, who would go on to win the Nobel Prize not very long after, was not allowed to have her own laboratory in Germany at that time, so only men could. And so they had to set it up together in order for her to be able to do this work that won her the Nobel Prize. Okay, so lots of different developmental EMTs, whether you're talking about in Drosophila or you're talking about in mammals. And now we start to have molecules that we can apply to looking at whether every EMT in an embryo utilizes the same kinds of control molecules that were identified as being important in Drosophila. And it turns out that they do. So the upstream signals, whether you're talking about gastrulation in a fly or gastrulation in a mouse, or neural crest migration, or palate morphogenesis, or tooth development, or development of the cardiac cushions, these are all important places where people study EMTs. they have different upstream signals that activate this process. And that's true in cancer as well. But what I want to tell you today is that all these upstream signals converge on a small handful of regulatory molecules that were the same ones that were found in that screen in Drosophila. Okay? So this kind of makes sense, because if you look back at the slide, and some of you have had cell biology where you've learned all the signal transduction pathways, we have tyrosine kinase receptor signaling, which can be activated by FGF. We have TGF-beta signaling. We have Wnt signaling. But there's really just a pocket full of these signaling pathways in embryos, and they have to do everything. And they're then reused during tissue homeostasis, and they're misregulated in disease. And so what that means is that they don't so much carry information as trigger an outcome in a cell that's already set up to respond in a particular way. And so you can trigger an EMT through many upstream signals, and that's not going to matter as well as the cell is set up so that the context it reads those signals, it says, turn on the factors that can make that transition from being an epithelial cell to being a mesenchymal cell. Okay, so what are those factors? Well, the ones that were found in Drosophila and then later in vertebrates that are most critical for both developmental EMTs and these pathological EMTs are shown here. Slug and snail, they're very closely related to one another and to an initial factor snail that was identified in Drosophila. Twist is the one I mentioned to you before. CYP1 is another one. We consider these to be the core EMT regulatory factors. They are misregulated in pretty much every cancer where it's been looked at, and they're implicated in almost every developmental or other kinds of pathological epithelial to mesenchymal transitions. And a characteristic of these guys is that you can introduce them into epithelial cells. This happens to be a breast cancer cell that's a non-invasive cell, and they're capable of mediating this transition. You can see that this cell has junctions with its neighbors, whereas this one has this different kind of polarity that I mentioned, a front-back polarity, and a phenotype where it's really poised for invasive behavior. Okay, so what's true in embryology is true in cancer. There are many different kinds of tumors out there, many different kinds of core kinds of tumors, but also different patient signatures. Not every patient who gets breast cancer is going to have the same molecular signature, and breast cancer is very different from leukemia, which is very different from some other cancer, colon cancer, okay? So there are many upstream signals that can mediate this process by which cells acquire this invasive behavior, but they all tend to tunnel down onto the same set of transcription factors that mediate this behavioral process in embryos as well. And that was the third historical thing that had to be realized to bring us to where we are in our understanding of tumor cell metastasis today, that these molecular regulators first found in Drosophila embryos and later shown to control this process in vertebrate embryos were actually misregulated during the process by which cancer cells leave a primary tumor and go elsewhere to form these metastases that are so deadly. And the first factor that this was really realized for was that one I mentioned, snail. Okay, snail was one of the very first factors shown to be important for gastrulation, that very early embryonic process. It's important for these neural crest cells that my lab study, and we use that as a model for understanding snail regulation and misregulation in my laboratory. But it was the first of these core EMT factors that was found to be present in invasive cancer cells, but not in non-invasive cancer cells. And I can give you a couple of examples of that. So this isn't as well labeled as it should be, I apologize, but these top three panels are normal breast tissue, okay? And this column here that you're looking at, that's that adhesion molecule, ecadherin, that helps those cells stick together when they're part of an epithelial layer, okay? And this middle column is looking at the expression in that tumor samples and in normal breast samples of this factor snail, okay, which was so important for those embryonic cells to be able to move. And in the normal breast cell, you see that these cells light up very nicely with this adhesion molecule because they're all part of an epithelium. They're all interacting with one another as a tissue layer, okay? But in these bottom panels, now you're talking about a tumor that has a grade where it's an invasive cancer. And what you can see sometimes is areas where this adhesion molecule has been lost. And if you look, and here you're looking at this, a slice through a tumor, one of the things that you can do is take an adjacent slice and look at whether another gene is being expressed. And this is an adjacent slice, and exactly where you've lost the expression of that adhesion molecule, you've upregulated the expression of this EMT regulatory factor. So now we're not talking about a Drosophila embryo, now we're talking about a breast cancer sample. And so many, although still not all clinicians, believe that a process of EMT occurs during tumor progression. I think pretty much universally, everyone believes now that whether you want to call it an EMT like there is in an embryo or not call it an EMT, that these transcription factors that regulate invasive behavior in an embryo also regulate the acquisition of invasiveness in cancer cells, at least of epithelial origins. One of the reasons why there is still some controversy is pathologists in particular are really, I need to see it to believe it kind of folks. And when you're talking about a transition whereby a tumor will shed a small number of cells to go elsewhere via the vasculature to make secondary metastases, it's not going to be usually this huge swath of cells that you see that are changing signatures of gene expression. And in fact, I have one more cancer sample for you, which is probably a more typical one. In this lobular carcinoma sample, it's just this scattering of small cells here are the snail positive cells, and these are actually cells within the stroma, so they've left the primary tumor and they're going to begin that process by which they go elsewhere to make those new tumors. Okay, you can also use cancer cell models to study the function of these factors, and a lot of work has been done on that, and a lot of work is being done here, both at Northwestern and elsewhere. And snail and those other factors that I mentioned are capable of taking cells that are non-invasive cancer cells and making them undergo this transition to an invasive phenotype. Okay, and that's equally true of these other factors as well. My laboratory studies all of these. We occasionally, when the mood strikes, study them in tumor cells, but oftentimes we use those embryonic processes because they give us a lot of control in terms of being able to do structure function type studies, take these molecules apart and ask how do they normally work, what goes wrong when they're not working properly, how could we maybe intervene in the process of metastasis, and that's what I'll end on in a moment. There are other ways that you can model using embryos the behaviors of tumor cells, and I just wanted to show you this one. It uses that other factor, twist. You can take advantage of chick embryos to do some very nice tumor invasion assays, where you can take fluorescently labeled, in this case, non-invasive breast cancer cells, and layer them on top of a membrane in the chicken embryo. And if they're non-invasive, like control ones are, they stay on the surface. But if we introduce this factor twist into them, they're now capable of invading down into the embryo and indeed going into the vasculature and circulating. We haven't let these guys go long enough to ask whether they can make tumors in a chicken. Okay, so lots of different upstream factors that can turn on a core set of regulatory molecules that can control this process. And I don't want to leave you with the idea that they're interchangeable. More and more data suggests that each one of these factors controls subsets of this process by which cells lose contact with their neighbors, acquire a mesenchymal phenotype, acquire the ability to invade the vasculature, et cetera. And that may, in fact, different balances between them may be why there are these partial EMT processes and not all EMTs look alike. There's a lot of work to be done trying to understand the unique and overlapping contributions that these different factors make to this very important process. And people are starting to take a systems level approach to this as well. I thought I would just throw this up here, but you can do all these connectivity studies now to try and figure out proteins that might have relationship to other proteins. So if you take these core EMT factors and computationally ask what other proteins are they associated with, either in functional assays or physical assays or so forth, you can use that to identify new proteins that might be important in tumor invasion and metastasis. That's been done. And by and large, when you do find things, either by looking at connections in the embryo samples or connections in tumor samples, you find it in the other process as well. So there's a lot more to this very complicated process than just these few transcription factors I'm telling you about. They regulate a lot of things, and metastasis can proceed in different ways, and there's a huge amount still to learn. But they really are, at the end of the day, the central regulators that get this process started. And so we like to use developmental models as a way of getting at or bettering our understanding of pathological EMTs. And I don't want to leave you with the idea that that's completely uncontroversial. There are some folks out there was a review published by a guy named Ragul Kalluri at Harvard a few years ago now, suggesting that EMTs developmental, pathological, and in fact different kinds of pathological EMTs. This is an EMT of a tumor cell. This is one that occurs during another disease called fibrosis. He suggested that each of these is so different from one another that one can't be used as a model for the other. I'm not sure I buy that. I actually think that, as I mentioned, different developmental EMTs are as different from one another as any one of them is from a tumor EMT, and different tumor EMTs can be as different from one another as they are from a developmental EMT. And so on some level, I think it's semantics. I think what we can say is at the end of the day, whether you want to call them one EMT process or 3 EMT processes or 300 EMT processes, they are controlled in some way by the same toolkit. And the job for the future is trying to take apart the toolkit and understand how each of the parts work. Okay, and so that means that while there's a lot of interesting work to be done up here, I'm really fascinated by what's going on in the nucleus by these transcription factors that control this process. Because I think, if you think about it, there's a lot of talk about personalized medicine, to go in and test your tumor to see what its unique signature is so that you can design therapeutics that are specifically designed for you that might not work on somebody else's cancer. But if at the end of the day what really matters is the ability to make metastases or new tumors, and if the core regulatory molecules that control that process are not as divergent but are really well conserved from tumor to tumor and patient to patient, then they should be an excellent model for therapeutics that would then work more universally. And so I'm going to very quickly tell you that a lot of the work that we do is on these 2 critters. We don't work on fruit flies, but we work on frogs and fish, believe it or not, because you can make lots of them in the laboratory, do lots of experiments. And the fish in particular, both of these develop very fast in a laboratory dish and are very amenable to molecular perturbations. But the fish is also transparent, so you can do all sorts of experiments where, for example, you could put fluorescent tumor cells in. People have done some nice work looking at tumor reprogramming in the microenvironment by injecting fluorescent cells into embryos. Okay, these are the cells that we study, these neural crest cells migrating through the embryo. And as I mentioned, the morphological behaviors that they undergo are just like the ones that tumor cells undergo. But more importantly, for the work that we do, these molecular mediators, these transcription factors, of which, again, I'm going to point out that slug and snail are a particular kind of transcription factor which is near and dear to Tom's heart because it coordinates metal ions in order to bind DNA. So they're called zinc finger transcription factors. They make a particular structure, because they coordinate a zinc ion. Okay, so these guys are related to one another and they have these zinc finger transcription factors. This guy here, CYP1 or ZEB1, it's a different family, but it also interacts with DNA targets using these same zinc fingers, and it binds to the same DNA sequence that these guys do. So all 3 of these factors, so 3 out of the 4 main players in metastasis, are zinc finger containing E-box binding factors. And then there's a 4th one called twist, which is not a zinc finger protein, it's a VHLH protein, but it also binds to these signatures called E-boxes. And so the fact that these factors are so important in all EMTs and in all tumor samples that have been looked at, and 3 of them are very similar to one another structurally and functionally in the sense that they interact with the same target site in DNA, suggested that they might be a good target for therapeutics that might be different from the ones that I was talking to you about before, things that shrink tumors or things that reduce the ability of tumors to attract the vasculature. Instead, we might be able to, by targeting them, go after this last part of Weinberg's puzzle, which is the ability of cells to leave the primary tumor and go elsewhere. So if we could just target as many of those core EMT factors as we could and turn them off, yes, we might still have tumor growth, but we have ways of dealing with that, including surgically. But we might be able to block the ability of these cells to leave the primary tumor and go elsewhere to make those metastases that are going to kill 95% of patients. Okay, so then we had these factors and wanted to have a way of going about that. And one of the great things about CLP and Northwestern and now the physical science oncology Center is it brings, dyed-in-the-wool life scientists like myself into contact with folks that come at problems from in different ways. And so just to give you a little bit of data at the end, I'm going to tell you briefly about a project that was a joint project between my lab and the Mead lab in the chemistry department here, and CLP and the Physical Science Oncology Center, that was spearheaded by an IBIS graduate student who spent time in both laboratories. So she really did exactly what this center is supposed to do: bridge the worlds, right? Bring the two sides together to do something that's greater than what either one of them could do individually. And so this was the work of a student, Allison Harney, who graduated not long ago and is now off doing a postdoc in New York. But what we set out to do was to target and inactivate those EMT transcription factors. And to do that, Allison built a tool that took advantage of two different aspects of these proteins. One was the fact that they need that zinc ion, and they fold in a particular way by coordinating it using histidines. Okay, and the Mead lab had a reagent, a cobalt shift base complex, that could modify those histidines so that they could no longer coordinate the zinc. And then we could take advantage of the fact that these factors had to bind in E-boxes. So we could put those two modalities together, and we could now target the cobalt shift base directly to factors that could bind the E-box. And if they happen to be zinc finger factors that had these histidines that were favorable targets, that should now make those structures unable to fold in the proper way, and therefore unable to bind DNA and regulate targets involved in invasion and metastasis. So that was the idea, and it had the additional benefit of being true. It actually worked, and that worked even better than we hoped it would, because it turned out to be temperature sensitive as well. So we could dial it up or dial it down, its activity, depending on whether we were at a permissive or a restrictive temperature. So this is just a model for how that works. Okay, if we don't have the compound around, or we're at a temperature where it doesn't function, these factors bind to target DNA, and they can regulate genes that are involved in this transition from the epithelial to mesenchymal and then ultimately invasive state. But if the compound is around, it would modify the DNA binding domain, prevent them from regulating their targets, and therefore prevent this EMT. And so I'll just show you two data slides before I end. This one showing that if we go into a cancer cell line, remember I told you that one of the things that these factors did was to dial down that cadherin, E-cadherin. As we put in increasing amounts of the compound, we restore E-cadherin expression, so we're knocking out the endogenous activity in cancer cells of these factors. And then I always like to look at it in embryos. So this is your only embryo picture that you have to look at. But these are little tiny frog embryos. And these stripes that you see are cells migrating from the top down towards the bottom, and you can see how far they've migrated. But then if we zap them with this compound, and again, this migration is dependent on those same factors, slug and snail and CYP1, those cells get stuck up at the top. They can't migrate, and that's just quantified over here. So no matter where we target, this is how much migration should have happened, and we knock that migration way down. And so by going after these commonalities in a process, these factors that get activated by diverse signals, but then in developmental and pathological EMTs are required to control all of those downstream aspects of the program, we can then prevent this transition in embryos, and now we're moving that into disease models. And so I'll end there by where I started. This transition from the epithelial to mesenchymal state is so important for both development and disease. And there's a bunch of really interesting places that this work has to go. What I didn't have time to mention is these same factors that are involved in this transition have more recently been implicated in the acquisition of stem cell attributes. So it may be that by undergoing an EMT and starting the metastatic process, you create a cancer stem cell, which now has the developmental potential to make a new tumor at remote sites. So understanding the link between these factors and stemness is important. Understanding the individual contributions of different EMT regulatory factors to this process, and understanding what controls when they do and do not carry out these processes are really important directions. But I will stop there, and the only two people that I really want to make sure to acknowledge is Allison, who did the work on the cobalt shift base stuff, the only data that I talked to you about, and she was a joint student with the Mead lab here at Northwestern. Thank you very much. In an adult. So if you were going to introduce this as a drug, you're looking for off-target effects that aren't in the tumor. That's a really great question. So in all of your tissues, you often have stem cell populations that are going to replenish adult tissues, and it does have the possibility of, in some of those cases, presenting these kinds of remodelings that would be important for that. And so what's true in all of cancer therapeutics, and that again is one focus of CLP and the PSOC, is targeting modalities, getting it to the right place at the right time. So we should probably move on, and we'll all be around, I think, to answer sort of joint questions at the end. And so our next speaker is going to be the inimitable Sharon Stack, who's back to join us and hopefully never to leave again. We didn't tell you that?
I have a flat tire when I go out.
Already took the spark plugs. Take it away, Sharon.
Give me just one second.